Dr. Sage here, back to continue our discussion of the eukaryotes, which we study in microbiology. In today's video, we're going to discuss the fungi. By the end of this video, you should be able to list 3 general features of fungal anatomy, differentiate among the terms heterotroph, saprobe, and parasite, explain the relationship between fungal hyphae and production of the mycelium, describe 2 ways in which fungal spores arise, and list 2 detrimental and 2 beneficial activities of fungi from the viewpoint of humans. The kingdom of fungi includes approximately 3 to 4 million species of fungi and can be divided into 2 main groups. Macroscopic fungi, things such as mushrooms, puffballs, and gill fungi, and microscopic fungi, things such as molds and yeasts. So, if we're talking about yeast, a yeast cell has a round oval shape and uses asexual reproduction. Hyphae are long thread-like cells found in filamentous fungi or molds, whereas a pseudohyphae is a chain of yeast. So we can see examples here of hyphae that have septates, which are the divisions between them, hyphae which are non-septated, so they don't have the divisions between them, and pseudohyphae, which are actually yeast cells, but they form long chains. And then we can have dimorphic, which is some fungi can take either form, the yeast individual cell form or the hyphae form. Here's an example of a dimorphic fungus that grows in soil exposed to bird feces or bat feces, which is guano. In the outdoors, it typically grows as a mycelium, but when the spores are inhaled, it responds to the high internal temperatures of humans by turning into a yeast that can multiply in the lungs, causing chronic lung disease. Now, recall that yeast are a eukaryotic cell, so they have the standard features of a eukaryotic cell. They have membrane-bound organelles, including things like the nucleus and the endoplasmic reticulum, rough and smooth ER. They have the Golgi apparatus. They have the mitochondria. And then they have things that all cells have, such as the ribosomes, the cytosol, the plasma membrane. Now, how do fungi get their nutrition or their source of energy? Well, they can be heterotrophs, which they acquire their nutrients from a wide variety of substrates, or saprobes, which obtain substrates from dead plants and animals, or parasites, in which they live on the bodies of living animals or plants. Most microscopic fungi grow in loose associations or colonies. A mycelium is the woven, intertwining mass of hyphae that makes up the body or colony of a mold. Septa are the crosswalls dividing hyphae into segments. So for example, let's say that we have a fungi growing on a piece of wood, like we would have here. Okay, if we zoom up, what we'll find is we'll see the mycelium. That's these white kind of fiber-like structures you can see here growing on the substrate, the brown wood in the background. If you remove the substrate, this is what the mycelium would look like. Okay, now if you zoom in on the mycelium, in particular on one strand, which is called a hyphae, this is what you would see. And the hyphae are divided by cross walls, these things kind of here, which are called the septa. Or we can have spores, which are the fungal reproductive bodies. So you have the hyphae here, for example, but this hyphae ends in a sporangium, which is a structure that produces spores. So for example, a mold that you can find on bread, which kind of looks like this fuzzy substance here. Okay, if you were to zoom in, what you would see is you have sporangia at the end of stalks of the hyphae, and these sporangia contain the spores of the fungi. Fungi can have many complex and successful reproductive strategies. For example, they can have simple outward growth of existing hyphae, and then they can have fragmentation, which is separated pieces of mycelium can generate a whole new colony. The primary reproductive mode, though, is spore formation. Now, of note, fungal spores should not be confused with the more resistant, non-reproductive endospores you find inside bacteria. So the endospores of bacteria and the spores of fungi are not the same thing. Fungi spores are used for reproduction. Bacterial endospores are not used for reproduction. So there are different types of fungi and different types of life cycles that the fungi can have. Some examples are zygomycete, asomycete, posadomycete. We're not going to go into those details in this video. But of note, they can alternate between spores and the hyphae forming the mycelia. So, the sporangiospores are formed by successive cleavages within a sporangium. But you could also have conidospores or conidia, which are free spores not enclosed by a spore-bearing sac. The purpose of sexual spores. Important variations occur when fungi of different genetic makeup combine their genetic material. Slight variations in the form and function are potentially advantageous to the adaptation and survival of the species. So how do we identify and cultivate fungi? Identification in medical specimens can be performed by isolation on special media, observation microscopically and macroscopically, asexual spore-forming structures and spores used to identify genus and species, and hyphal type, colony texture, pigmentation, physiological characteristics, and genetic makeup. These are all methods we can use to identify which species the fungi is. Most human fungal infection occurs through accidental contact with the soil, water, or dust. How do you get exposed to pathogenic fungi? Well, community-acquired infections caused by environmental pathogens could be one cause. Hospital-associated infections caused by fungal pathogens in clinical settings, or opportunistic infections caused by pathogens infecting already weakened individuals. For example, those who have a weakened immune system. Other medical conditions caused by fungi can be allergies or neurological conditions due to toxin production. But fungi are also beneficial to us. They serve an essential role in decomposing organic material and returning essential nutrients to the soil. They also form stable associations with plant roots that increase water and nutrient absorption. For example, this is a plant root covered by mycelia of a fungus, but this fungus is helping the plant by helping its absorption of nutrients. We as humans also use fungi to help us produce antibiotics, alcohol, organic acids, and vitamins, as food flavoring, and we use them to produce alcohol in beer and wine, and gas that causes bread to rise. So, this was your brief introduction to the fungi, one type of eukaryote that we study in microbiology. Until next time, this has been Dr. Sage.